Alzheimer's genetics mapped risk alleles that condemn a brain, but this leaves ambiguity separating cause from wreckage. If we invert the question and search for protective alleles present from conception, the logic tightens. The arrow of time ensures the defense came first. Every protective allele is a natural clinical trial. The genetics prove where a pathway is held. To read this ledger of defense accurately, we grade these genetic factors on a strict evidentiary scale, from Tier 1 established proofs down to Tier 4 conjectural observations. The genome defends the brain across 5 distinct biological layers, reducing the toxic substrate, clearing the debris, sharpening the immune responder, fortifying the target synapse, and slowing the tempo of aging. By mapping the highest graded defenses across these 5 layers, we can locate the exact biological joints where Alzheimer's disease is physically severable. We are not searching for theoretical interventions. We are reading the exact biological steps that have already spared human minds. The most absolute defense operates at the beginning of the cascade. This is the substrate layer, where genetic resistance prevents the toxic pathology from accumulating in the first place. The most decisive Tier 1 example comes from a massive genome sequencing project in Iceland, which uncovered a rare mutation in the amyloid precursor protein. A single amino acid substitution, alanine to threonine, slightly alters the protein shape. This mutation reduces beta-secretase cleavage and lowers amyloid production by roughly 40% across the carrier's entire life. Clinical trials attempted to mimic this by developing BACE1 inhibitor drugs like verabecestat. They failed. Administering a massive, near-total inhibition of the secretase enzyme to an aging brain worsened cognition rather than protecting it. The Icelandic variant demonstrates that copying a genetic defense requires matching its exact tuning. The protective biology relies on a fractional, lifelong calibration, not a late-stage biological hammer. Moving to the middle of the cascade, we find the brain's innate immune cells. Microglia are built to contain pathology, but as Alzheimer's progresses, they often fail, inflaming the tissue around the plaques. The PLCG2 P522R variant is a Tier 1 defense that acts as a hypermorph. It slightly amplifies the cell's internal signaling, turning the microglial response up and making the cell a more efficient clearer of debris. A different defense, the Tier 2 CD33 variant, achieves the exact same clearance goal through the opposite mechanism. It lowers an inhibitory receptor on the cell surface, releasing a biological brake to unleash the phagocyte. These genetic instructions do not silence the immune system or delete the inflammatory response. They simply equip the cell to handle the physical burden of the disease. The innate immune system operates as a tunable rheostat. The genome provides multiple distinct pathways to reach the identical protective setting. Resistance stops pathology. Resilience is entirely different. It means preserving the mind even as the physical lesions of the disease heavily accumulate. The most extreme human proofs of resilience exist in an extended family in the Colombian department of Antioquia. Thousands of relatives carry a fully penetrant mutation that guarantees early onset dementia by their mid-40s. 2 individuals in this kindred possessed rare genetic deviations, one carrying the APOE3 Christchurch variant, the other the Relin-Colbos variant. Both evaded the family's schedule, maintaining intact cognition for 3 additional decades. Scans of their brains revealed an enormous burden of amyloid plaques. The disease pathology ran completely unchecked, yet their memory and cognition survived. Both variants operated on the exact same cellular bridge, weakening the binding between ApoE and heparan sulfate receptors. A 2024 study showed this protection scaled across 27 other relatives, effectively decoupling the amyloid burden from the spread of tau. Because this mechanism spared the brain while amyloid ran free, it identifies the induction and spread of tau, not the amyloid itself, as the disease's proximate executioner. When mapping these factors onto a strength-graded ledger, 2 principles dictate the architecture of resistance. First, the strongest defenses cluster at the extremes. They work by making less toxic substrate early, or by sparing tau pathology late. Almost nothing targets the amyloid plaques themselves. Second, the defenses are the exact mirror of the risks. APOE4 increases risk, APOE2 lowers it. An APP valine mutation guarantees early dementia, an APP threonine mutation prevents it entirely. The genome does not carry a separate defense program. It carries a single set of biological dials, and protection is simply the outcome when those dials are turned in the correct direction. Every protective allele identifies a potential drug target and explicitly dictates the direction a therapeutic must push the pathway. But the Tier 1 defenses carry a strict translational barrier. They are fixed in the germline from conception. We cannot retroactively install them in patients. The only installable defenses lie in the epigenome, the chemical marks that regulate gene expression, like sirtuin activity or the restraint of HDAC2, can be rewritten across a lifetime. They are reversible. These epigenetic markers sit lower on our evidentiary scale because they are vulnerable to reverse causation. We still have to prove whether the mark actually protects the brain or merely reflects the presence of the disease. This creates the strategic logic for the field to use the causal authority of the fixed defenses to identify the targets and pursue the plasticity of the reversible defenses to actually hit them. The therapeutic future of Alzheimer's disease does not rely on inventing novel biological mechanisms. It relies on learning how to pharmacologically imitate the exact biological settings of the people who never got it.